and at least had a cup of coffee or something to eat. And there is apples and cookies and carrots from Rebecca's farm on the dining room table. So please go and help yourself whenever you want. A couple of housekeeping-y things. Are there people here who weren't here yesterday? I think there's a couple. Ginny, Drew, and Paco. Yeah, Paco, right. Paco is our translator, and the person that was coming yesterday was called suddenly back to Mexico where there was a death in their family. But we've got somebody who's stepped in, so that's really great. That all happened overnight. The other housekeeping thing is that tonight, as you know, when the meetings finish at 4:30, you're on your own until we get to the public meeting at 7. But there are a couple of things if you don't want to be on your own. We've got a sign-up sheet for a restaurant which is actually right in the building of the Y, where the auditorium is for the public meeting. It's an Italian restaurant, and we've booked some spots, and we've got a sign-up sheet there. We just have to let them know by noon who would like to go there. It's a pizza, pasta, $15 a shot type place, but there is wine and beer and stuff like that. The other alternative is, since we do have leftovers from yesterday, and we may well have leftovers from lunch, who knows, which is coming today, we're going to do a quick buffet supper here for people who would like to have an early supper here at, well, when the meeting finishes, 5:30, so that you're down there by 7 if you would like to eat here. But again, we'd like to have an idea of numbers. So we're asking people to sign up. There's another sheet out there. And the third option is there's all kinds of restaurants between here and the Y on 87 Elm Street. So just walk down, take the subway down to Dundas, walk along Dundas a couple of blocks, and you'll come across Chinese, Korean, Japanese, Thai. You won't come across a McDonald's or anything like that, but you'll come across all these little restaurants. So, the Duke of York pub. Okay, yeah, that's just down the street. And there's a craft beer place in the basement. So if you prefer craft beer, you just go out here down to Prince Albert, and there's a pub right there.
At the corner of Bedford and Prince Arthur.
Yes. So these are all your opportunities. Now, another point to get to the public meeting tonight, you can walk. It's a half an hour walk just straight south to Dundas and then a little bit east. But there are, for those who don't want to rummage around and figure out where to get change for the TTC, there are tokens on the table out there and toonies. If you're young, you take a token. So that's the little one. So just remember that, little for young. And if you're old, you take a toonie, and that's the bigger one. And that gets you one way down on the subway. So if you feel it's a toonie and 10 cents really, but I mean there's no 10 cents, there's just the toonies. Old is 65 and over. So there you go. I can't remember if there's anything else that notes. If you do have people who are presenting or who've made any kind of presentation, we would really like to get notes electronically or your presentations. Saturday will be when we are talking about what we're doing following on from this, but if we don't have any notes or any PowerPoints or anything that was said here, then we'll be relying on our memory and we might just make it up. I know that some people have been taking notes, but I noticed that one of our best note takers isn't here yet, but hopefully he's coming. Okay, I think we're nearly ready. We're going to start with an opening reflection and prayer from Richard Schneider. So I'm going to turn it over to Richard, and then we'll move into our first panel. You want to sit there? Or if you stand up, Richard, it's better to explain to the people who are
use the mic, please.
Use the mic. Oh, I can use the mic. Oh. To explain to the people who are here, we're live streaming this thing, so that's what's going on at the back. But people need to stand up and use the mic so that you can be really seen on the cameras and wherever it is that it's been live streamed to. Do we have any evidence that anybody's looked at this?
There's 14 views overnight.
Very good. From Canada or from?
I didn't look, to be honest.
Okay.
I was reflecting on all those challenging and excellent, I was reflecting on all those challenging and excellent questions that Peter was writing on the paper yesterday, and thinking, what does one say in the face of that? And the answer actually came from going into the washroom here. Because when you enter the washroom, it's still dark in there, but the first thing you walk past is a ceiling-to-floor mirror. So what you see in the dark is yourself. But you don't think it's yourself, you think it's someone else. It's quite startling.
You'll have to choose the other washroom next.
And where that led me was to this. So we're going to start with a little dialogue between Job and God. I'm going to start in the middle of this. Job has just challenged God, really shaking his fist under his nose, actually, saying, these people are arrogant, this is all pompous, do something to them and justify me. Right? Give me justice because I have suffered so much. And this is the middle of God's answer. Behold Behemoth, which I made, as I made you. He eats grass like an ox. Behold, his strength is in his loins, and his power in the muscles of his belly. He makes his tail stiff like a cedar. The sinews of his thighs are knit together. His bones are tubes of bronze, his limbs like bars of iron. He is the first of the works of God. Let him who made him bring near his sword. For the mountains yield food for him, where all the wild beasts play. Under the lotus plants he lies, in the covert of the reeds, and in the marsh. For his shade the lotus trees cover him, the willows of the brooks surround him. Behold, if the river is turbulent, he is not frightened. He is confident though Jordan rushes against his mouth. Can one take him with hooks, or pierce his nose with a snare? Reflecting on that, Job answered the Lord, I know that thou canst do all things, and that no purpose of thine can be thwarted. Who is this that hides counsel without knowledge? Therefore I have uttered what I did not understand, things too wonderful for me, which I did not speak. Hear, and I will speak. I will question you, and you declare to me. I had heard of thee by the hearing of the ear, but now my eye sees thee. Therefore I despise myself and repent in dust and ashes. We were cautioned yesterday that the way to become like that is to listen to the wisdom of the elders. Well, for the topic we're going to be talking about in this symposium, at least a good portion of the elders are right here in this room. And the problem is that we're coming here with our disparate knowledge and our disparate languages that come from our disparate disciplines. And the question for all of us is what's bringing us into this one room and sitting us down together? And part of the answer is in that humility once Job actually hears, right, what God has to say about what the world is. So I'll end this by reading a psalm. It's the 103rd psalm or the 104th psalm, depends on your numbering. In the Orthodox Church, it's a daily prayer. Right. Bless the Lord, O my soul. O Lord my God, thou art very great. Thou art clothed with honor and majesty, who coverest thyself with light as with a garment, who has stretched out the heavens like a tent, who has laid the beams of thy chambers on the water, who makest the clouds thy chariot, who ridest on the wings of the wind, who makest the winds thy messengers, fire and flame thy ministers. Thou didst set the earth on its foundations, so that it should never be shaken. Thou didst cover it with the deep as with a garment. The waters stood above the mountains. At thy rebuke they fled. At the sound of thy thunder they took to flight. The mountains rose, the valleys sank down to the place which thou didst appoint for them. Thou didst set a bound which they should not pass, so that they might not again cover the earth. Thou makest springs gush forth in the valleys. They flow between the hills. They give drink to every beast of the field. The wild asses quench their thirst. By them the birds of the air have their habitation. They sing among the branches. From the lofty abode thou waterest the mountains. The earth is satisfied with the fruit of thy work. Thou dost cause the grass to grow for the cattle and plants for man to cultivate, that he may bring forth food from the earth and wine to gladden the heart of man, to make his face shine, and bread to strengthen man's heart. The trees of the Lord are watered abundantly, the cedars of Lebanon which he planted. In them the birds build their nests. The stork has her home in the fir trees. The high mountains are for the wild goats. The rocks are a refuge for the badgers. Thou hast made the moon to mark the seasons. The sun knows its time for setting. Thou makest darkness, and it is night, when all the beasts of the forest creep forth. The young lions roar for their prey, seeking their food from God. When the sun rises, they get them away and go down to their dens. Man goes forth to his work and to his labor until the evening. Lord, how manifold are thy works. In wisdom hast thou made them all. The earth is full of thy creatures. Yonder is the sea, great and wide, which teems with things innumerable, living things both small and great. There go the ships, and Leviathan, which thou didst form to sport in it. These all look to thee, to give them their food in due season. When thou givest it to them, they gather it up. When thou openest thy hand, they are filled with good things. When thou hidest thy face, they are dismayed. When thou takest away their breath, they die and return to their dust. When thou sendest forth thy spirit, they are created, and thou renewest the face of the ground. May the glory of the Lord endure forever. May the Lord rejoice in his works, who looks on the earth and it trembles, and touches the mountains and they smoke. I will sing to the Lord as long as I live. I will sing praise to my God while I have being. May my meditation be pleasing to him, for I rejoice in the Lord. Let sinners be consumed from the earth, and let the wicked be no more. Bless the Lord, O my soul. Praise the Lord. So we were also cautioned yesterday that we should always keep in our minds that life and all the scientific and theological problems of life are caught up in this vast and complex intersecting web of the connection of all things. In order to remember that, though, we must remember it like Job, that in fact, we are the food for the lions that God has provided. And if we approach our problems in that spirit, then as wise elders, the Lord will open our lips and our mouth will sing forth his praise. Amen.
Thank you, Richard. I just wanted to read one sentence, and it's from the front page of the Globe and Mail today, but the headline is bizarre. Top court deals blow to Indigenous peoples. And this is what's going on in Canada, that the Indigenous peoples have lost their right to protect their lands in BC and it's now going to be turned into a ski resort. And that's what is going on in Canada, and it just reflects, I think, on what we were talking about last night. Anyway, we're going to move on today to looking at synthetic biology. And our first panel is a couple of people who are well known in the field, and we're really privileged to have them. There's more here who are well known in the field, but we're starting with two, Jim Thomas and Craig. So the first person is Jim. Jim is going to speak first and show some slides, and then Craig, and then we're going to have a conversation briefly between the two of them, and then with you. So, Jim, you're going to speak from there, or yeah, you're going to come up here. Okay. Jim is the research program manager at Etc. So if you don't know Etc., check it out on Google. It's one of these mighty little groups. He's a writer. His background is in communications, writing on emerging technologies and international campaigning. He has extensive experience on issues around transgenic crops, nanotechnology, and synthetic biology. He was trained as a historian to look back at history of technology, and now he's looking forward to the future as to where is this technology going. So we're really pleased that we've got Jim with us, so I'm going to hand it over to Jim.
Thank you very much, Anne. Does this sound all right from that? Yeah. I really appreciate this this gathering amongst the elders. I was very pleased to discover that according to the local metro, I'm a young person. That's wonderful. I think my task and particularly thank you, Anne. I'm sure it will be said a number of times this weekend, the time and effort and attention that Anne and others in the organizing team have put into bringing this to this place has been really quite inspiring. So thank you, Anne. I think my task to kick off today is to try and present what is this object, this kind of knot of meaning, synthetic biology. As Anne said, I work with the Etc. group. We're sometimes called a sort of a technology watchdog. We work beyond just technology. It's also around biodiversity and food and farmers' rights and so forth. The heart of what we do is we track emerging technologies, strategies in corporate concentration, what companies are doing strategically, to try and understand trends on behalf of our interest is what this means for the most marginalized people in society, for peasant farmers, for indigenous peoples, for peoples who are workers, disabled people, and so forth. So that's the focus of what we do, is trying to see how these emerging trends impact the most marginal. And for the last decade or so, I've been with others in my organization watching this field of synthetic biology, writing about synthetic biology. Here are some of our publications. And as I say, I think I'm supposed to answer this question in a kind of common way. We have a common understanding of what is synthetic biology. And I'm very conscious that in this room we have somebody who, this is Drew, for some people who've met. Drew Endy is one of the people who really brought together this notion of synthetic biology. And I find myself, every time I'm asked to define synthetic biology, going back to a conversation with Drew, next one, which happened at the President's Commission on Bioethics. I don't know if you remember this, where this was 2011, something like that, and the two leaders of the President's Commission were asked. Drew was on the stand actually, and it was sort of the question here is, I have 5 definitions of synthetic biology. People are saying that it's must exist in digital code and be processed in the genome, that it's just an extension of genetic engineering, but then someone says there's no distinction with genetic engineering, and Amy Gutman, who was the head of the President's Commission, said yes, and somebody told us if you'd had 5 synthetic biologists in the room, you'd get 6 definitions. And then they asked Drew, can we have another one, please? And Drew's answer was instructive. I refuse. And don't stress out about it. And I think that's fair enough. Over the years, I've found it very difficult to define synthetic biology. It's not necessarily helpful to have a perfect definition. However, this is a definition that was developed by the United Nations, by the Convention on Biological Diversity, and there's a small expert group which Fred is on, I'm on, and others. And we tried to come to a definition. We were asked to come to a definition by the 196 countries of the CBD. And so we provided this, and it got adopted last year. It's a further development, a new dimension of modern biotechnology. This is a continuation, rather than a different thing from genetic engineering. It's part of genetic engineering. It goes further. It's about the design, the redesign, the manufacture, the modification of genetic materials, living organisms, and biological systems. And what you might notice is this is a tremendously broad definition. Pretty much anything that's genetic engineering fits in this, and that's kind of the point. Next one. Then in fact, I find a more useful place to start is this graph that was in a report that was produced for the Department of Commerce about 10 years ago now, US Department of Commerce, which, as you can see, basically shows, there's a set of techniques, roughly call them transgenics, recombinant DNA, that were the way in which we did genetic engineering. And there's a new bunch of techniques, let's call them synthetic biology, biological standard parts, simplified operating systems, and so forth, that's also now being used to do genetic engineering. And that sometime around 2010, one set of techniques has pretty much taken over from the other set of techniques as the way in which we genetically engineer organisms. And that one of the implications of that is that you can do it faster, quicker, with more intervention. And that this inflection point for biological technology is really what synthetic biology is about, that we've reached an inflection point, a change, an intensification in some ways in how we engineer living organisms. Next one. That genetic engineering ain't what it used to be, that something's changed. The next one. And I think to grasp what's changed, of course, you need to have a sense of what it was, what our father's genetic engineering was, and now what our children's genetic engineering is. Let's say GMO 1.0, what came out of the 1970s is, roughly speaking, we might call it transgenics. This is basically a cut and paste approach to the genetic systems in organisms. Next one. And I just want to, first lay out the sort of metaphorical understandings of this approach to genetic engineering. And I say this very tentatively because I know Craig's going to rip this to pieces in his next presentation. But anyway, the notion that underlies genetic engineering is actually what's called the central dogma of genetics. The idea being that DNA, this molecule, which is made up of these 4 base chemicals, G, T, C, and A. These chemicals, depending on how they're ordered, create a code. And that code effectively instructs the organism on how it develops, how it produces particular proteins, how it expresses itself. It's rather like this DNA is a kind of long ticker tape, a sort of molecular ticker tape, which has a kind of an instructions hardwired into it, and that you're effectively running this ticker tape through the cell. That's kind of the metaphor here, in which the cell is kind of the processing machine, in a way. So in classic genetic engineering, the way in which you would change the way in which an organism behaves is you get to this ticker tape, you get to the DNA, and you look through it. There's our gentleman genetic engineer there looking through the DNA code, and find particular parts of instructions, and then you'd cut them out, and you'd move them somewhere else. You'd find the instruction for creating a red protein, or the instruction for creating a green glowing protein, and you'd cut it out of a jellyfish, and you'd put it into a potato, and the potato would glow green. It's kind of cut and paste. And how would you do that? Well, there were, roughly speaking, 2 major techniques. One is that you would having cut that out, you'd put it on a little gold bullet and you'd literally fire it into the DNA of another organism. That's the gene gun approach. Or you would get a bacterium, Agrobacterium tumefaciens, to smuggle it into the DNA of another organism. These were the ways in which you did recombinant DNA, transgenics, roughly speaking. So what's changed? Well, first of all, our access to those codes, the DNA codes, has expanded enormously. We now have, there's the gentleman on the left, and now there's a woman. Now we have the ability to read large amounts of genomic data, those genomic codes. So there's around 1,000 large-scale genome processing centers around the world. Next one. And the quantity of genomic data that we have is really ballooning, the information about those codes. So if each of those letters of genomic data was a grain of sand, what we currently have is the equivalent of about a grain silo worth of genomic data. And that's 32,000 microbial genomes, 5,000 plant and animal genomes, 250,000 human genomes. That's quite a lot to work with already. But if you look forward, the predictions are this is really going to explode. From one paper, it's assumed that there would be 1,000 million trillion base pairs by 2025. And as you've put there, that's the equivalent of over 150 stadia full of grains of sand. And at that point, we have the genomic codes for all 1.2 million known species of plants and animals in databases. And that's basically, at that point, a new industrial raw material for doing genetic engineering. That's the raw material. That's what's available. Next one. Additionally, we have a set of new tools. There were those two tools I showed first. Now we have really more and more ways of changing the genome, changing DNA, not just DNA, RNA, and other genetic molecules. Next one. So you know, we can attack the genome in various ways. We can change its code in various ways. There's a list of some of these, and this is really not a very good list. It's just what came off the top of my head. Next one. And then, thirdly, there's a move to try and make genetic engineering actually engineering. And Drew, actually, and his colleagues have been a major part of this. That what was being done before wasn't engineering. It was a kind of experimental, artisanal process. That real engineers work with predictable parts. They work with standard substrates. So there is an approach to say, those little bits of instructions, can we make them standard, repeatable instructions that you can use again and again, like you have instructions in a programming code or the parts of an electric circuit. And so you have the creation of these biobricks or standard parts. This idea of trying to make natural systems more engineerable out of standard parts is nothing new. Engineers have been doing this for centuries, beyond centuries, actually. Really, wood has been made into these standard parts. We call them planks, and makes it easier to design as a result. Stones made into standard parts, blocks, and that again makes it easier to design complex, interesting things. And so the idea being, if you can make genetic systems into standard parts, then you can design more quickly and more reliably complex and interesting things, like this glowing down there. Next one. And a further change, it's not really a change, but it's just an intensification, is that whereas it always used to be that you would go looking for the DNA you wanted somewhere in nature, you can build it now. You've always been able to build it, but it's now able to build it much more quickly. That the synthesis of DNA, the making DNA as a synthetic chemical has become very standard. You could buy something like a DNA synthesizer on eBay. It doesn't cost you very much. It's got those 4 bottles. And then you could say, I want a piece of DNA that goes GTTCATC, and it would print it out. But you don't need to. You can go to DNA 2.0 or GNAT or one of these DNA synthesis companies and give them your credit card and say, I want a piece of DNA that goes GTTCATGCTCGG, whatever. And each one of those letters, they'll charge you, it's like 5 cents, 7 cents, something like that. And that would be sent to you by post. So it means that, if you go to the next slide, it means that, that the ability to write DNA is now becoming a driving force in how we do genetic engineering. This is from Rob Carlson last year, and I'm sure this is very out of date already. He's saying in this that there's currently around 1 billion base pairs of synthetic DNA, gene length synthetic DNA being printed every year. Currently about 4.8 billion base pairs of those letters, or bases of those letters. Roughly equivalent to the size of one human genome every year being printed. And at around 7 to 17 cents per base pair for the gene length, or these oligos, these short single-stranded DNAs at about 5 cents. The point being, this is going to keep coming down in price and up in speed. The next one? Sorry, the next one. Yeah. Yeah. Yeah, sure. So just as the ability to read DNA is speeding up, the ability to write DNA is speeding up. That if you want to build a piece of DNA chemically, you would order from a DNA synthesis company. It would cost you about 7 cents per letter of DNA, and they would send it through the post to you. And we now have an international industry, which is in aggregate printing something like 6 billion, roughly, letters of DNA every year, and sending it around the world to labs to use. And so that is, just for sense, that's roughly speaking about the size of a full human genome that's being printed every year. So the blue line is actually sequencing, how the cost of sequencing is dropping. The red line there is short. These oligos, they're single-stranded DNA and they're very short. They're easier to make. And then gene-length DNA, which is double-stranded, is the yellow there. So it's showing that that's going down too. But the next slide, I mean, just an example, if you were to make the full genome of polio, polio is not a living organism, it's a virus, 7,500 base pairs, work it out, it's about $500 roughly. You could order a synthetic version of polio by post for about $500. Now, hopefully, no company's going to send you polio in the post. Hopefully, they won't even send you the parts of polio in the post. They have detection to try and pick that up, hopefully. But it's doable, right? This is something that's very simple to do. And it has been done several times. But what we are seeing at the moment is a new project called HGP Write, Human Genome Project Write, to develop the technologies to synthesize on the scale of human genomes. That's to say 3 billion base pairs. You could do that now. Roughly speaking, by those numbers that Rob Carlson gave, you're looking at about $21 billion for a human genome. That's about a third of a Zuckerberg. That's about a fifth of a Bezos. That's about a quarter of a Gates. It's about a week of US military spending. It could be done, so it's a lot. But the idea is to bring this down to some sort of price and speed that is actually commercially feasible. And this process, the HGP Write, has companies like Autodesk behind it who would like to get genome synthesis massively increased. Next one, sorry. And then, of course, you don't actually need to necessarily print out new DNA strands. You could go into an existing DNA strand and just re-edit it. This is the area of genome editing. You'll have heard of CRISPR, which has received a lot of press. But there's also zinc fingers, talons, direct mutagenesis, a number of other approaches, which basically go into existing strands of DNA and cut out little parts, put in little parts, change little things, where you're going in and changing the DNA sequence sort of in place. Does that make sense? Yeah, okay, next. And there's an example of some of those. Next one. And probably I think what's going to become increasingly significant is you don't really need humans to do any of this. The bots can do it. That you have these robotic construction facilities. So for a while, it's been true that you can have, you can tell a robot to do the actual pipetting and so forth for genetic engineering. But increasingly also the question of design is being handed over to artificial intelligence algorithms, machine learning algorithms. So you have companies like Zymergen, where artificial intelligence algorithms will work out 1,000 or so changes that they think will come to a particular outcome and then implement them in a microbe. Not just one microbe, maybe thousands of microbes in parallel, and then screen through those. So increasingly, this is not being done by individual gentlemen genetic engineers in their labs. It's being done by brute force machine learning algorithms with robots. Next. And together what this means is that you have the emergence of genetic modification as a commodity scale. Something where you have large amounts of data as the underlying raw material. You have this robotic automated genetic engineering. You have these tools which can do so. And this becomes a commodity scale service. Next. Sorry, I keep putting these in. Underlying this is the idea that you can program the life forms to do various things through this commodity scale genetic engineering. And life becomes, synthetic life becomes this sort of digital platform where you're changing the codes, remixing the codes, programming the codes. And that is driving, for example, this. iGEM, again, Drew's very involved and can talk more about this, is an international competition that happens every year. It has, I don't know, how many teams this year, Drew? So 6,000 people. These are undergraduates and high school students who work with standard parts of synthetic DNA to design organisms. They're not called organisms, they're called genetically engineered machines. This is a mechanical engineering approach. And to come up with interesting, useful, cool uses for a genetically engineered organism. The next. And of course we have an industry that will call itself, and sometimes the synthetic biology industry, sometimes doesn't use that term. Claims for how this industry is growing. There's claims that it's going to be a $40 billion industry by 2020. I think that's overheated, but it may well be like a $16 billion industry, something in that order. We're seeing a lot of government funds, particularly defense funds. In the US, the largest funder, in fact, probably the largest government funder worldwide, is DARPA, the Defense Advanced Research Projects Agency. Many Fortune 500 companies who are using this technology, and I'll talk a minute how. Rapid expansion into venture capital, and so forth. If I look at this industry, I see sort of three waves, really. And they're not waves, they come at the same time, but it's a way of expressing that. The first is an industrial approach to biosynthesis. I'm going to describe that in some detail in a minute. The other is really going beyond the GMO approach, particularly in agriculture, new ways of manipulating crops and animals in agriculture. And the third is, I'm calling it here molecular computing or molecular communication, using DNA as a substrate for communication. We'll come to that in a second. So biosynthesis, this is basically making stuff in vats using genetically engineered organisms, synthetic organisms. Next. So to get your head around this, think about how yeast makes wine or beer. And if you can imagine that yeast as being a kind of a machine, a factory, that takes sugar and turns it into alcohol. Next. Now, if you could re-engineer with a little bit of genetic code that machine, you could reprogram it with the codes that would, rather than producing alcohol, produce vanilla or vanillin, the key compound, or the green glowing protein that made that green glowing bunny that I showed you. And you then re-engineered using the tools of synthetic biology with that. The next thing you would have is basically a reprogrammed factory that did that. It would be a tiny little yeast cell that was producing, amongst other things, a small amount of vanillin or a small amount of that protein. And a small amount isn't very much when you feed it sugar. But the thing about these is these are living factories. They reproduce. So you can make many thousands and millions of these, and you can put them all in a large vat. And that, once you scale it up into a fermentation vat, is a way to make vanilla or vanillin or plastics or flavors or fragrances or so forth. And you can then bring those compounds to market. This is basically the biosynthesis industrial business plan. Next. And so off this business plan, the synthetic biology industry first began looking at what are the largest markets? Fuels, chemicals. And the initial few $billion going into this industry were coming from Exxon, DuPont, Shell, who were trying to make biofuels at a time when the oil price was very high. And probably about 2/3 of that initial funding was trying to come up with things like biofuels. But then the oil price dropped, and there was a recognition that this wasn't a very good way to make money, that things like fuels and plastics and industrial chemicals require high volumes. You need to be able to ferment in high volumes, and you get very little value off of it. It's pennies per liter. And so the search went for, can we find things that can be made in small volumes, but are worth a lot more? And that took to things like flavors, fragrances, cosmetic compounds. Next one. Particularly what are called secondary metabolites in plants, the spices, the oils, and so forth. And really one of the projects that led this direction is this. It's the artemisinin project by Amaris and Jay Kiesling. And they worked out the sequences that would be needed to re-engineer a yeast to produce artemisinic acid. Artemisinic acid is a precursor to artemisinin, which is an anti-malarial compound, usually comes from the wormwood plant, from Artemisia annua that you see at the top there. And so rather than making artemisinin, this important anti-malarial from Artemisia annua, you would make it from a genetically engineered yeast in a vat. Next. But what drove this was not so much, and it was partly humanitarian interest in creating an anti-malarial and making sure there's lots of them, but actually the question was initially, how could we make one of these, isoprenoids? It's a class of chemicals that are found in nature. There's 75,000 of them. How could we make one of these isoprenoids in a way that once you'd made one, you could change the genetic code and make a different one, and then change it again and make a different one? What they were trying to do at Amaris, what Jay Kiesling was trying to do, was come up with a platform for producing any one of the 75,000 isoprenoids. And they happened to find that the Gates Foundation would give them $52 million to do this if they went after an anti-malarial. And having done so, they then built a company on making one after another isoprenoid compound. And overall, there's around 200,000 natural compounds that you could potentially get to. There's something like roughly 8 different metabolic pathways, broadly speaking. And if you can engineer each of these metabolic pathways, theoretically, you could get to any one of those 200,000 natural compounds. You would have a living machine that could get you to whatever it is you want at the end. And so that's the business plan for a number of these biosynthesis companies. Here's Evolva. You see in the middle their pathways, and these are some of the final products that they claim they can get to. Stevia, cocoa, turmeric, chili powder, musk, breast milk, and so forth. That by altering the genetic pathways within yeast, they can get to a range of natural products. There is potential for biosynthetic routes to completely replace any natural sources, says Caleb Kirsch of Lux Research. That's a pretty bold claim. But that's what this industry is claiming theoretically it can do. And you have companies like Amaris who are now offering, they call the Pathways Program, to sponsor and secure a molecule. If you're a flavor and fragrance company or a cosmetics company and there's a particular natural molecule that you want to produce, you go to Amaris and using their algorithms, they'll design for you an organism that will make that compound. That's the explore biosynthetic production opportunities with minimal risk and commitment. Next. And so what we now have is an industry that's mostly producing flavors, fragrances, cosmetic ingredients, supplement ingredients, food ingredients, soaps. And the big players now are not so much the oil and gas and chemical companies, although they're still involved, but who's most excited, I see now, is the flavor and fragrance industry. This is an industry dominated by top 4 companies who control 57% of the flavor and fragrance market. And all of these companies are working on synthetic biology, flavors, fragrances, spices, and so forth. And they're interested in synthetic biology for a few reasons. Firstly, at the moment, they currently source a lot of their flavors and fragrances from farmers. From small farmers around the world. It's a very broken up market. It's very difficult to be on top of all those different supply chains. If you have a single large vat, you can make the world's patchouli production or the world's vanillin production in one very large vat or one facility. And so the idea of, and you're not stuck with weather problems and hurricanes and so forth. So the idea that you have a reliable supply is very attractive. Secondly, it turns out that because this is a fermentation process, legally, you can call this natural. Anything that's fermented under US and European law is considered natural. So you can say this is natural. Well, this is what's claimed.
There's no legal definition for natural in the US.
Yeah, there's no legal definition, but this is what the companies will claim. Yeah.
So that's what the FDA claims.
Right. But the companies will claim that they can call this natural, and they are calling it natural. And thirdly, price changes, obviously. If you can produce large amounts in a vat of sugar, or maybe methane, increasingly this is moving over to natural gas, maybe you could do so far cheaper than you would have done if you had farmers picking it in the field and trying to send it by boat and so forth. Next. And I just want to draw attention to this, that at the moment this industry relies heavily on small-scale farmers. 20 million small-scale farmers and agricultural workers produce the botanical crops that feed into this industry. That's who's going to be affected by this. 95% of the crops used in the flavors and fragrance industry are grown by small-scale farmers, mostly in the global south. That's who's going to get replaced if this changes en masse. Okay, next. I touched on this point about what's natural, what you can call natural. For companies, the ability to call this natural is a great advantage in the marketplace. The word natural on a food or a cosmetic gets a higher bump in the market than the word organic. So if they can get away with it, that's a lot of money. Of course, the implications are, what does that mean for those who grow truly natural botanical ingredients? We'll talk about that later. I'm very glad that Alejandrino's here, who can talk much more about vanilla and what that means for the forest and for peoples. I thought this was just a very telling quote. Terravia were producing a algae oil that they could structure to replace cocoa butter. And this is somebody from Terravia saying, we can dislocate the production of that oil from a tropical climate to the middle of Iowa in winter. It's truly revolutionary. We can take what's a normal crop cycle and compress it into 3 days. Or as Max Chaxko from SymBioBeta, which is a trade group for the small synthetic biology companies, step aside cocoa farms, synthetic biology is on its way. The good news is Terravia actually just went into receivership. So this isn't happening as fast as they might have hoped. But you see the issue. Next. We recently produced a report looking at 13 case studies of how this impacts different areas where natural products are grown by farmers, and we've produced this natural products map where you can explore which countries might be impacted if there was to be a shift from natural products to synthetically produced natural products. Next. And also in a database. This is at database.symbiowatch.org, where we were trying to understand which ingredients are coming to the marketplace that are synthetically produced. We found about 350 entries. Around 75 seem to already be on the market. Another 75 seem to be promised for the market in the near term, and the rest it's very unclear. And I'm sure there's a lot more. It's very hard to dig into this area. Next. I mentioned this question of feedstock. I'm just mentioning it in passing. It's not like producing something in a vat has no effects on feedstock. You need sugar. Potentially, if you're making something like oil or biofuels, you might need a lot of sugar. So this has real impacts. And increasingly, companies are moving over to methane, to natural gas as the feedstock. And that has implications for the development of fracking and taking natural gas off oil fields. There's real world effects there too. And then, separate from biosynthesis is this area of what I'm calling GMO 2.0. That some of these same techniques, particularly gene editing, are now being applied to crops, to animals, to insects, to produce a next wave of genetically modified organisms for food and feed and so forth. In Europe, but also now elsewhere, the industry likes to refer to these as new plant breeding techniques or new breeding techniques. And has led a discussion saying that these new breeding techniques are not really GMOs. They're different from GMOs. They're put together differently. And therefore, they should sit outside of GMO regulations. They're talking about CRISPR, zinc fingers, talons, and so forth. They'll argue that because you can go into the DNA and you can change a letter one at a time, you've got nothing new coming in. It's not transgenics. There's nothing coming from elsewhere. And therefore, the public's not really so worried. And therefore, we don't need to label it or regulate it so much. civil society is very much involved in a fight around this, saying, no, this is still engineering the genome. You're still changing the sequence. It may, in fact, be more significant. And so the big fight right now is this GMO or is this not GMO? GMO is a 4-letter word. Next. But we're seeing here Monsanto, DuPont, Syngenta too, others who are using these technologies as a quicker way of modifying corn or of modifying commodity crops. It's an extension of what's already been happening, but faster breeding. And in fact, we already have at least one product in the market that's gene edited. It's a very small change. It's just a couple of deletions, really. But it's a herbicide-resistant canola that's already sold. And it's sold as non-transgenic. The CBUS make a big point, and they actually get farmers get extra money because this is non-transgenic. Next. And that's significant, although in fact the non-GMO project considers it as GMO. So this fight over what's GMO or not, and I just put this up, this is work that's going on modifying bananas, Cavendish bananas, through gene editing to resist a fungus. It's a very important project if you believe in having large monoculture banana production. But see how it's presented. Gene-edited crops bypass biosafety regulation. If we can use this technology, we can get around all the regulations. We can get around the oversight. Next. And I just thought we should wave a flag for something that's coming. It's not genetically modified organisms, but the use of synthetic RNA in RNAi sprays. So here is where these little strands of RNA, the messenger molecule, is being sprayed across a crop. If it gets ingested as a spray, if it gets ingested by an insect, it then interferes with the genetic functioning of that insect. There's no organism here. Once again, it's not a genetically modified organism. Okay. And I want to finally finish on one of the uses of synthetic biology, gene editing, that I think we have to discuss, and it's very significant, and that's gene drives. And I'm just going to take a few seconds to describe gene drives, because I think we're going to discuss this a lot. So a gene drive is an element, a genetic element, that if you were to use it to genetically modify an organism, it would spread a trait through a population. Now, what do I mean by that? If you genetically engineer an organism, let's say to glow green, used that example a couple of times already, so I'll stay with that, and you were to release that organism into the wild, let's say it's a fly, theoretically, about half of its offspring might pick up that trait, and half of its offspring might pick up that trait, and half of its offspring, and theoretically over time that trait would disappear into the background. That could change, of course, with evolutionary pressures, but that's the idea. If you put a gene drive in, this synthetically engineered construct, it means that every single one of its offspring will have that trait. They'll all glow green. And every one of its offspring will glow green. And every one of its offspring will glow green. Until over several generations, you've taken over the entire population, and the entire population glows green. It's a way of driving a particular genetically engineered trait through an entire natural population. It was called the mutagenic chain reaction. It was really only proven a few years ago by Ethan Beer at San Diego. And he did this with fruit flies. He genetically engineered a fruit fly with a gene that would turn the fruit fly kind of pale yellow, and a gene drive. He put them in a box full of fruit flies, let them go a few generations, and when he opened up the box, only yellow fruit flies came out. As he said there, we were stunned. It was like the sun rose in the west rather than the east. It overturns an assumption about Mendelian genetics inheritance. It showed that you can drive a trait through a population. I find that terrifying, I should say right up front. The implication, of course, of Gantz and Beer's experiment was if one of those fruit flies had got out of the lab in San Diego and had mated with fruit flies around the lab in San Diego, you may have seen spreading yellow fruit flies throughout Southern California, throughout the US. Where does it stop? Potentially, does that change all fruit flies everywhere? Potentially. There's some reasons why that might not work, but that's potentially what you're talking about. But other people find this extremely exciting. So GBIRD is a project of a conservation organization island conservation and also the US government. It's funded by about $3.5 million of funds from DARPA, the Defense Advanced Research Projects Agency. Why DARPA is funding conservation? That's a whole different discussion. And the idea here is to release gene-drive mice onto island, and these mice are called daughterless mice. These mice will only, when they mate with other mice, will only give rise to males. And when they mate with other mice, they'll only give rise to males, and so forth and so on, until there are only males in the population, at which point the whole population collapses. So this idea of the daughterless mouse as a way of destroying populations of rodents on islands that are in themselves invasives and destroying bird populations is seen as a way of supporting conservation. And this consortium is now looking for somewhere to release these daughterless mice. Western Australia, New Zealand, Hawaii, it's looking at different places. A similar project has been a proposal to do this with mosquitoes, in Hawaii specifically. There's a mosquito, Culex, which carries an avian form of malaria, which is responsible for the demise, the upcoming demise, really, of this very beautiful honey creeper bird. It's retreated to the mountaintops because of industrial agriculture in Hawaii, and now the mosquito is decimating its numbers. What if you could release gene-drive mosquitoes that would destroy the population of Culex in Hawaii? That would save the honey creeper bird. That's the suggestion. And of course, the question of, can you use this to stop malaria? Can you use this to stop schistosomiasis? Can you use this to stop Zika, human vector-borne diseases, is very much top of mind. You have Target Malaria, which has about $75 million from the Gates Foundation. It's looking in 3 African countries, Burkina Faso, Uganda, and I forget the third. Oh, Mali, yes. To start doing the work for potentially releasing gene-drive mosquitoes that would destroy the mosquitoes that carry malaria. You can see the attraction to the Gates Foundation, but there's many reasons why this is problematic. There's also suggestions to release these into agriculture to make weeds that are resistant to Roundup no longer resistant to Roundup. Monsanto likes this idea, and so on. This is a statement from a number of conservation leaders. Nemo is in the middle there. Given the obvious dangers of irretrievably releasing genocidal genes into the natural world and the moral implications of taking such action, we call for a halt to all proposals for the use of gene-drive technologies, but especially in conservation. This is going to be a huge fight in the coming year, especially, whether gene drives can move into experimentation. And this is an experiment on the scale of evolution that is moving forward very quickly. And I'll stop there. Thank you.
I'm sure people have all kinds of questions, but I want to, just stand up and turn around and say something to your neighbor if you've got a question, and then we're going to sit down, and then we're going to have Craig, and then we're going to have a bit of a discussion, then coffee, and then we're coming back into a lot of more discussion. So please stand up, turn around, shake a leg, and say hello to your neighbor. What did you think of that? Well, in one sentence. Okay. Okay, can we settle down again? There will be opportunities for more discussion. I know everybody's bursting with questions. I just find this is just, what in the world can we do? Anyway, okay, let's move on. Maybe by the end of tomorrow we'll have figured out something that we can do. Are we ready? Craig, you going to speak from here?
Yeah.
Okay. I'd like to introduce our next speaker to you. Can we all settle back down, please? If you need to go out and pee or get coffee, please do, but there will be a break in half an hour. 20 minutes, well, we better. Okay, our next speaker is Craig Holdridge. Craig is the director of the Nature Institute. He spearheaded the founding of the Nature Institute in 1998, which is in Ghent, New York. He is author of a book called The Question of Genes and the Giraffe's Long Neck. That's another book, The Giraffe's Long Neck. Craig teaches and lectures all over the place, including in here, but all over the world. His approach is to learn from nature. He tells the story of plants and animals as dynamic and integrated beings within the larger web of life. So I know we're looking forward to hearing what Craig has to say and build on what Jim has to say, and then there'll be coffee, and then there'll be talking. Oh, you're over there.
Thank you very much. I hope you can still pay attention for a little bit. Jim presented us with an amazing amount of information in a short period of time. And what I would like to do is just to present some other pictures that complement what he brought. And I think, in reflecting on what was said last night, and just about the whole topic of synthetic biology and genetic modification of organisms, that I thought it would be important that we don't only talk about the technologies and have mainly the technologies themselves in the foreground, and the background for all the technologies, or the medium for them, is our living organisms, right? The living organisms are what we're manipulating, what we're changing, what we're viewing. So I thought, in a way, simple way, and I hope accessible way, to give you an impression of the way I think organisms live in the world. And I just want to give you a couple of concrete examples, and then we'll move on from there. So, first slide. That's not quite sharp, is it? Is it? Anyway, it's good, I guess good enough. It's a little bit weak, but it's all right. This is a plant, as you all know. And plants develop out of seeds. And when a plant develops out of a seed, it develops over time, and every part, the roots, the stems, where the side branches, the leaves, the flowers, the fruits, and again, the next generation of seeds, develop all in relation to each other. No part develops out of some pre-existing part that is already of the same kind. They develop out of embryonic tissue, and when the roots develop, they are making possible that the leaves can do photosynthesis. When the leaves begin doing photosynthesis, they make possible that the roots can develop further, and so forth. So that the plants are in continual activity to grow and develop as long as they live. So we can say a plant develops out of a seed. Next. That's a seed of this plant. It's called wall lettuce. It's a little tiny seed, or fruit, actually. And when we say a plant develops out of a seed, we're actually saying something that's not quite correct. Because nothing that comes later, that was in the slide before, well, you can move on, which is the same slide again, is in the seed. That is all a new creation. That is a creation, you can say, of the plant, but it's not a creation only of the plant. It's a creation of the activity of the plant in relation to an environment. And I won't go into all details, but at least we can say light, air, water, and minerals. Without that world, without that context, the plant, of course, could not become what it is. It is by virtue of its relation to the world. And that relation becomes very vivid when you start looking at plants growing in different contexts. I want to give you one example. Next slide. Wow, that's really light, almost invisible, but it is a plant. And it's the plant of the same species. Both of those are wall lettuce specimens. And what's the difference? Well, we can say it easily. It's a different environment. The one plant, the one on the left, was in a shadier environment, and the plant on the right was in a more sunny environment. And so a greater light intensity. And of course, the plant has roots, it has leaves, it has stems, it has flowers, it has seeds, both cases. But every aspect of the plant on the right in detail is different from the one on the left, looked at from the other side. Namely, look at the forms of the leaves, for example, the degree of rooting and branching of the roots, the coloration, you can see it in the stem, different pigments have formed. The plant on the left on the right has much thicker leaves, has a more woody stem, formed many more side branches and many more flowers. And so when you go into the details, you have the first impression, but then you go into the details, you begin to see, well, there's what we call the same in an abstract way, leaves, but in detail, those leaves are different. And where's the difference come from? Where's the difference come from? The difference comes from the way this organism has interacted with what we call the environment, meaning the environment, so to speak, becomes embodied in the plant through the activity of the plant in relating to that environment. It's a very important thing to realize how connected, deeply connected, intimately connected the plant is with the environment, to the degree that when we're talking about a plant, we're talking about an activity that's expressing the qualities of its environment. It's not a thing unto itself. That does not exist. That's a mere abstraction. It's a mere abstraction. And we do have lots of abstractions in our heads that guide our actions. So this is why I think it's fundamental or important to look at some very basic but very fundamental qualities of organismic life. So a plant, if you think back to the seeds, I think next slide, go back to the seed. Imagine that seed now could be planted in very, very many different conditions. There are infinite possibilities of expressions of wall lettuce, because in every context, it will meet a different situation. And of course, each seed has in some form a different inherited background. So you could say in this sense, we know it's going to stay the same species if something strange doesn't happen. But in every case, you have a plant that is creating itself in relation to that special situation and is one of a kind. All right? So I'll come back to some of this later. So that's something that I've studied a lot and have, I could say I've gained a great respect for what plants can do. And I've become to see that plants are fundamentally activities and fundamentally plastic in the sense of have what we more abstractly call phenotypic plasticity, a great degree of ability to modify themselves at any time in relation to changing circumstances. And that's all the way down. And I don't want to go into the all the way down. That would be another half an hour to go into the all the way down. But that's when you study all the literature on genetics and epigenetics today. It just confirms that basically what you're seeing here, if you consider a molecule, which you can't look at in this way, but you can experimentally work with, you find the same thing. A molecule that looks like it has this particular function in this context, that's true. You change the context, it's doing something different in a different way. All the way down, you have flexibility, plasticity, context, sensitivity, the ability to change, and that change is integrated into the whole of the organism, because the whole plant changes, not just a part. One other example, a very different one. which I don't want to show the slides yet. This is from an animal, and it's a drastic example, but it shows something also fundamental. In the 1940s, in a agricultural research farm in Holland, a little goat was born. And this little goat, I mean, there were many goats born, but this one was special because this goat had no front legs. So it was malformed. Nobody knows why, what brought that forth, as a proximal cause, no idea. It was a phenomenon that was there. And this goat seemed healthy, and they let it live. They didn't say this is abnormal and let it sacrifice it, kill it so that it doesn't suffer. Because the goat was very soon hopping around on its rear legs like a kangaroo, except it doesn't have the long tail of a kangaroo to rest on, right? So that didn't develop. And so this goat lived a somewhat strange but happy bipedal goat life for about a year until there was an accident and it died. And an anatomist at the university there studied its morphology. Next slide. And at the top you have the form of the spine of the bipedal goat, and in the bottom the form of the spine of the four-legged normal goat. And all the bones were there, right? The typical number, the typical arrangement of the spine were there. But every bone was different. And every bone was different in relation to the bone before it and after it. And of course, the differences in the bones are related to the differences in the way the muscles of this animal were being used. And that the fact that this animal, for some reason or other, and I don't know how to put it without being anthropomorphic, is to say it somehow got the idea, well, I can stand on two and I'm going to do this. But I don't think it actually got the idea. It did it and it lived, right? And in every aspect of its morphology, it shifted its bones, its muscular structure, its activity shifted in relation to the challenge that was presented by having this malformation. No part remained the same. One other slide. So that's a picture on the left of the normal rear leg and in the bipedal goat. Again, all the bones are there, but every bone in its, in this case, in its larger structural elements, but then also in the microstructure, changed as an adaptation to the activity of the organism, which was a response to a challenge that was presented to it. So this is something, I said, this is an extreme example, but what's good about it is it shows what actually is happening all the time in all animals, is that every animal, and we can think of ourselves in this respect too, that through activity, we are forming and changing the body that was inherited as a kind of model that then gets sculpted through our activity in our lives. Right? So that this shows us that when we're looking at an organism, like now I'll say like an animal, then the intentionality of its being to move in a particular way, to respond to certain kinds of situations, also to respond novelly to a situation it hasn't encountered before. That flexibility of behavior leads to different kinds of activities. Those activities change it as a whole organism in more gross ways and in very subtle ways. And again, all the way down. That's what one's finding with all the epigenetics research today, but which has been known phenotypically for a long, long time. Right? A lot of these things that one talks about in epigenetics today have actually been known for a long time. It's just that now it's becoming part of the forefront of genetics, where one's talking about context sensitivity, flexibility, crosstalk, etc., at the level of genetics, which you find in the phenomena themselves when you look at them at a macro scale, which is easier for us to understand at first. So, fine and good. Now, what does this have to do? You can move on, which means we can turn it off. With synthetic biology. So why was I doing this? Why was I talking about this? Because I wanted to give, you could say, the living quality of organisms a certain voice in this conference, right? So, okay, of what nature are we here? What am I as a living organism? And when we then shift to the perspective of synthetic biology, I don't need to go into a lot now because Jim brought it all, but if you just think of the terms or what I'm talking about now is a way of viewing, a way of thinking that motivates the research. So to redesign, to rewire, to reprogram by constructing parts, devices, modules, systems into organisms such that these function in highly controlled and predictable ways to produce predictable outcomes, right? So, but just redesigning, rewiring, reprogramming, all the metaphors that were in Jim's presentation, we need to be conscious about them. Okay, with what mind, what frame of mind are we approaching these organisms, whether it's yeast now or a plant or an animal or a human being, with what conceptual background, with what opinions are we working, or with what worldview are we working that we're bringing into the organisms, conceptually and then factually afterwards. So to keep this short, I just want to express this in the following way. Well, what do the organisms say to that? Right? What do they say to that? And I was thinking about this morning, and I said, well, here's an answer. And this is quick, and, anyway, I don't have much time. Since I am not a machine, and I have no parts, in the sense of synthetic biology, right? Devices, circuits, modules, and systems. Since I have none of those, in the way you engineers conceive of them, then if you want to manipulate me according to those ideas, then you are going to have to suppress or remove aspects of my plastic integration. You're going to have to suppress or remove my ability to momentarily adapt in novel ways to my changing environment. You're going to have to suppress my openness and sensitivity to changing contexts. That means you're going to have to make me ever more into the machine that you're envisioning from the very beginning. You're not creating life, you're changing organisms to be less than the living because you want to have standardized parts, modules that have clear-cut pathways where there's not disturbing crosstalk. You need to be able to have that high degree of predictability. And in order to have that, you have to change me in a way that does not actually correspond to many of the living qualities of me as an organism. So this is something that's well known. And so let me just read a quotation from 2015. In biological engineering, unfortunately, the progress has been hampered by the fact that the salient properties of both basic parts and functional molecules depend on their context, which includes the surrounding parts and modules. So unfortunately, these are context dependent. Another quotation, engineered biological circuits rarely work as designed. In most cases, the performance of their molecular parts is highly dependent on cellular and sequence context and varies greatly from one system to a next. So these are experiences that synthetic biologists have had. And it's interesting then to look and say, well, what's the answer to that? What's the response to that? And let me read you a couple of responses. This is from Batchelor and Collins, 2012. Synthetic biology urgently requires strategies to limit such context dependence. Right? So you have to limit it. What I was saying before, you have to, if we're looking to have more independence of parts and modules so that with the predictability, then we have to limit the context nature of the organism, to put it briefly. So, a more crass statement, an alternative to understanding complexity is to get rid of it, right? Or, from George Church, who many of you have heard of in the context of synthetic biology, you focus on the parts of science that you do understand and clean out the parts that you don't understand, right? And that, one can understand that. But the fact is, we're now dealing not with wood or metal, but we're dealing with plants, animals, of course, also microorganisms, think of the yeast. And the question is that, what kind of a stance is that? Right? And what are we doing when we are trying to manipulate something, and in so doing, we're cleaning out the parts that we don't understand in order to make it do what we want it to do. And you see, this is where, for me, the consideration of organisms and their characteristics moves into ethics, right? This question was there last night. It doesn't move into, it's implicit in it, because all science, all inquiry is a relationship to something. We are already in relation. The fiction that one learns about as value-free science is a fiction because everything is in relationship, and as a human being, I have responsibility for every relationship I go into. Or I can say, I just ignore that fact. But I take up a relationship, and there is an ethical component within that from the very beginning. So for me, at least the question should be asked, or could be asked, well, let me put it this way so I can come to a close. That when we study another living organism, one way to do it, another being on the planet, or other beings, if we look at it ecologically, then for me, the first question is, well, who are you? What are your characteristics? What do you have to show me about the nature of your life and your connections with everything else? I'm interested in that as someone who would like to know more about the planet of which I am part. And then it is another move to say, okay, now I would like you to do X, Y, or Z for me. Right? I would like you to do something, the utilitarianism. And of course, we do this as human beings all the time. And I think we just need to be very thoughtful about how we do that and whether we are taking the characteristics of the world that we're manipulating in a fuller way into account, or whether we rely on a more simplified view that can be very powerful and that can work to change life, as we've seen, and to at least be circumspect and say, well, this will have consequences. Because what I'm doing is, as I'm looking in a very narrow fashion and working in a very narrow fashion with something which is much bigger, more complex, more of the nature of activity than what I have in my head. And we will, I think, probably the technologies will get better and better, which means that the organisms will be forced to adapt to those more and more. And that's the question of, well, what do we want as human beings, right? I think I'll leave it at that. I know I could go on longer, but that's enough. Thank you.
Thank you. Thank you, Craig. You've given us so much, both presentations, so much to think about. And it is coffee time, and rather than open it up to questions, there's going to be a whole point afterwards where we're going to be having little what did we call it? Coffee dialogue tables, and that's where, and these people will be part of it. So I'd like to say now, do you want to say something about the, okay, so Peter's going to say something about the coffee dialogue, and then we're going to go for coffee or tea.
And before you go out and empty your mind of all that incredible presentation, take a moment and with the post-it notes that are in the center of the table, jot down the powerful, meaningful, relevant questions that are front of mind for you now. One question per card, and on the way out to the coffee, place them on the flip chart sheet here, and we'll work with them during the break. Jot down the powerful, meaningful, relevant questions that are front of mind for you now. One question per card on the post-it note, and on your way out to coffee, post it on the flip chart. Is the task clear? Okay, thank you. Thank you so much, and so feel free to head out to your coffee break when you're ready. And just one other quick announcement. Members of the Planning Committee who would like to review those questions together with me during the break or just at the end of the break, please come back and join us around a table here.